Uh, Welcome back, friends. In today's session we are going to continue with our topic of gaseous exchange and respiration. So today we have our session 3 in respiration topic, and today we are going to discuss about the respiratory substrate and the structure of mitochondria. So respiratory substrate ni zile substances ambazo tunakuwa tunazitumia kwenye respiration. So by definition, respiratory substances are those organic substances or food substances that upon being oxidized in respiration pathway, they yield energy in the form of ATP. So an organic substance which can be oxidized and by the oxidation reaction can yield ATP is what we can call the respiratory substrate. Now these include carbohydrate, fatty and proteins. So we have 3 major respiratory substrates as I described here. They are carbohydrates, fats and the proteins. So we'll start explaining about carbohydrates. So we are starting our discussion by discussing about the carbohydrates. When these are available, they are usually used first by most cells. So carbohydrates, more especially, it is present in a storage form in plants and animals. In animal, polysaccharides such as starch it is in plant, and in the animal it is glycogen. So glycogen is what we call it the animal similarity or the animal anomaly of the starch which is present in plants. And glycogen is also present in fungi. So they are hydrolyzed to monosaccharide before the interrespiratory pathway. We'll discuss about the respiratory substrates later again, and we shall see how carbohydrates it is doing, or it enters respiration, and how other substrates they are doing respiration. So in terms of carbohydrates, glucose it's the exact sugar which enters respiratory pathway. Glucose is the one which enters respiratory pathway. And when we are moving to another respiratory substrate, which is lipid, in the lipids we have fats and oils, as how we discussed in biochemistry. And the lipids, they are used mainly when carbohydrates they are reserved, have been exhausted. So if the carbohydrate which is stored in the body, it is exhausted, that means the body learns through deficiency of carbohydrate, then lipids they start to be used. So they are first converted to glycerol and fatty acids. First converted to glycerol and fatty acids. Fatty acids are energy rich, and some cell, such as skeletal muscle cell in particular, gain some of their energy in this way during normal activity. So during normal activity, hata kama mtu ana excess of carbohydrate, some of the cells, such as the cells of muscles, they use lipids because of the high energy which is present in the fat acids. So we shall see later how fatty acid they undergo what you call the beta oxidation, and by beta oxidation of fatty acids we are getting energy from them. And from there we have proteins. About proteins they are made up of amino acids. So we have the acidic group and the basic group in the amino acid. So in respiration of protein, the amino acid first we are doing deamination. We are removing the amine group, and by removing the amine group we are forming ammonia. That is, the reaction is taking place in the liver, and we call it deamination. Then from there we are getting the ammonia, and then we are leaving the keto group. So the keto group will go to enter the respiratory pathway and will be metabolized to release energy. We shall see later how this metabolism of protein or respiration of protein takes place. So since protein have other essential functions, protein have many functions in the body, so it is the last substrate to be used in the body. After the exhaustion of carbohydrate and the lipid, then we can use proteins. So kama mtu amefanya fasting for many days, then we are starting to use carbohydrate store. After using carbohydrate store, we are starting to convert lipids into carbohydrates by using different enzymes and different metabolic pathway. We can convert lipids into carbohydrate, and or another way we can use lipids in the we can hydrolyze them to get glycerol and fatty acids, and then the fatty acids, they can enter they can be broken down by beta oxidation, and they can enter the carbon cycle. But if the lipids they are exhausted also, we can use proteins. So hata kuna baadhi ya watu wanaenda kwa madaktari sio nini, anatafuta dawa kupunguza unene. Most of people ambao they are fat, it's not because of muscles, that their muscles they have heavier muscles, or it's not because of the atrophy of the hypertrophy of the muscles, because when you are talking of hypertrophy, we mean kwa mfano wale wanaonyanyua vyuma, muscles they undergo hypertrophy because they increase in size. So there is increase in size of each muscle cell. However, there is no increase in the number of muscle cells. So watu wengi ambao ni wanene, kama tu ule mtu hafanyi exercise kama za kunyanyua chuma, so ule unene unakuwa it's because of accumulation of fat. So the best method ili yule mtu aweze kupunguza unene, no need ya kwenda kwa daktari kusema nahitaji dawa kupunguza unene and things like that. The easiest way and the simplest one ni yule mtu kwenda tu kuanza fasting. So anaanza fasting during the period of prolonged starvation, au during the period ambayo atakuwa carbohydrate imeisha. Yule mtu anatakiwa tu anafanya fasting, asile chakula, au tu angalau anakunywa tu maji, then anaweza kuwa anafanya hivyo for 2-3 days, then anakula chakula kidogo, au hata anakula tu kidogo carbohydrate tu kidogo, let's say tea, labda na andazi moja. So at the end of the day, carbohydrate ikiwa exhausted in the body, yule mtu ataanza kutumia nini? Fat. So that is the time ambao mtu utakuta tayari anaanza kabisa kukonda baadaye, fat ikiwa exhausted. Lakini kwenda kutumia protein kwa mtu kama ambaye yuko fat inakuwa ni very difficult, kwa sababu anakuwa tayari ana large store of fat. Now let's go to discuss about the structure of mitochondria. The structure of mitochondria. So I know, because mitochondria is the main organelle which is used in respiration, and when we are talking of aerobic respiration, it must involve mitochondria. So I know this discussion about mitochondria will be longer, and just stay tuned. If you want a pause, you can pause the video and then you can continue, start learning about the mitochondria. So mitochondria, respiration takes place in the mitochondria. Mitochondria is the plural, in singular we call it as mitochondrion. So mitochondria, they are present in all eukaryotic cells, and they are major sites of aerobic respiration within cells. So they are present in all eukaryotic cells, and they are major sites of respiration in all cells. Major sites of respiration in all cells. Now I want us to move to the next page, so that we can proceed with our discussion about mitochondria. Now what we are saying is that the number of mitochondria varies from one cell to another. So the number of mitochondria per cell varies considerably, and it depends on the type of organism and the nature of cell. Number of mitochondria varies because different types of cells, they require different amount of energy stored to perform different activities. For example, cells which perform active transport, for example the cells of small intestine, they use active transport to absorb food substance from the lumen of the intestine to the bloodstream. Especially here we are talking about glucose and proteins, not lipids, because the mechanism of transport of lipids is somehow different from the glucose and proteins. Now, if the cells, or the columnar epithelial cells of the intestine, small intestine, they require large number of mitochondria. Why? It's because they require large amount of ATP to perform absorption work. Also, when we are moving to the cells of muscles, they require large amount of energy for movement, because contraction of muscles requires ATP. Contraction actin in the myosin filaments, they require ATP. When you are moving to liver cells, which we call them hepatocytes, they also require a large amount of ATP. So all in all, you are coming back to realize that it requires different types of cells. They require different amount of mitochondria so that to synthesize a variable amount of ATP per cell. Now the number of mitochondria per cell varies considerably and depends on the type of organism and nature of cell. Cells with the high energy requirements possess a large number of mitochondria. For example, we have liver cell containing upward of 1000 mitochondria per cell. Resty, less active cell possess far fewer number of mitochondria. Mitochondrial shape and size also tremendously variable. So the mitochondrial shape and size also it is variable. That is because we are in need of taking in the raw materials for respiration, and we are in need of removing out the end product of respiration. So that's why the variability of the shape of mitochondria help you to take in the raw materials of respiration and to remove the end product of respiration. So they may be spiral, they may be spherical, they may be elongated, or they may be cup-shaped. We shall see later, even in the topic of comparative studies of groups of living organisms, some of the living organisms, they have spiral mitochondria. So, yeah, but most of the time it is spherical and elongated. So, even branched sometimes, and usually large in active cells than in less active ones. So the shape of mitochondria will be large in active cells as compared with the less active cells. Their length ranges from 1.5 to 10 micrometer and width from 0.25 to 1 micrometer. But their diameter does not exceed 1 micrometer. You see, their length varies, width varies, but diameter does not exceed 1 micrometer. Now, what is the reason behind why the length and width vary but the diameter still remain the same? What is the reason behind? What could happen if the diameter could decrease? What could happen if the diameter could increase? Now, there is this question in the BS book. Why should the diameter of mitochondria remain fairly constant when the length is so variable? Why? What is the reason behind? Now, the reason behind is to increase the surface area for rapid diffusion between cytoplasm and mitochondria. As I've said, that there is the exchange of the raw materials and the end product of respiration from mitochondria to cytoplasm and from cytoplasm to mitochondria. So that exchange is through diffusion process, and in order for diffusion process to occur, diffusion requires large surface area to volume ratio. So diameter of mitochondria remain fairly constant, enable the large surface area to volume ratio to be maintained for the exchange of materials. Now, we are saying that mitochondria, they are able to change shape, and some are able to move from one area in the cell where a lot of activity is taking place. So they are able to move from one area in the cell to another area through the cytoskeleton. So cytoskeleton provides the means through which organelles they move within the cell, as how we studied in cytology. So this provides the cell with a large concentration of mitochondria in area where ATP needed is greater. Other mitochondria have a more fixed position, as in the insect flight muscle. So some of the mitochondria, they have fixed position, while others they can move from one place to another. So now let's move to see the structure of mitochondria. Let's see the structure of mitochondria, how each mitochondria can be seen, in the electronic microscope, because we know in the simple microscope or in the light microscope we can't see clearly the structure of each mitochondria, so we can't explain the structure of mitochondria based on the how it can be seen in the light microscope. So we have the page which explains the structure of mitochondria, and we'll discuss it. But this is how can be seen in the diagram. So I want to show you some of the structure, so that if we will go to discuss or to explain what I'm going to explain, you must not wonder what I'm explaining about. Now, here we have the outer membrane of mitochondria. This is the outer membrane of mitochondria, and it's the one which maintains the shape of mitochondria. From there we have the inner membrane. Inner membrane, it consists of the infoldings. As you can see, it consists of the multiple infoldings. These infoldings, they are called as the crista. Crista is the singular, but the plural is the cristae. Cristae. So all of these in combination we call them as cristae. But cristae, they have what we call the stocked particles. Stocked particles. Stocked particles, they are integrated with the ATPs responsible for the synthesis of ATP during the electron transport chain. So all of these they are different adaptations for mitochondria to perform its function. For example, these cristae, they increase the surface area for electron transport chain to take place. And as I told you that the stocked particles, they are packed with the ATPase enzymes, and the ATPase enzyme is responsible for synthesis of ATP during the end stages of electron transport chain, is how we shall discuss later. So here we have the space between the outer and the inner membrane. It is called the intermembrane space. It is called as intermembrane space. We discussed about the structure of mitochondria and cytology, but this is just like a repetition. Here we have the matrix. Matrix, it is made up of the enzymes responsible for the carbon cycle, because the carbon cycle, the Krebs cycle, Krebs cycle takes place here, and we have different enzymes which are responsible for stages of the Krebs cycle. We have the free 7th ribosomes, and then you have the circular DNA. Presence of the 7th ribosomes and the circular DNA account for the prokaryotic nature of mitochondria. So mitochondria is somehow prokaryotic in nature because of the presence of the 7th ribosomes, similar to the 7th ribosomes present in the prokaryotic cell, and the presence of the circular DNA. So mitochondria, they are organelles which are present in the cell, and they are able to replicate themselves because of the presence of the DNA, which is genetic material, and the circular, the small ribosomes, which is the protein synthesizing machinery of mitochondria. So they are able to synthesize their own enzymes, and that's why sometimes they are called the cell within a cell. But the explanation about how the mitochondria is regarded the cell within a cell, it is explained in the cytology topic. So you can view my cytology lectures so that you can know what is going on. Now, each mitochondrion is bonded by two membranes, which are called as envelope, as how I've shown you in this diagram, that these two membranes, the outer and the inner one, they are called the mitochondria envelope. So each mitochondria is bonded by two membranes, and they are called as the envelope. So the outer one being separated from the inner one by the narrow space, which is called the intermembranal space. Now the inner membrane is folded inwardly into a number of shelf-like cristae. Inner membrane is folded inward, as how I shown you in the previous diagram. Now the cristae tend to increase its surface area, providing the space for the components of respiratory chain which are located in the membrane. So cristae, as I told you, it tend to increase the surface area for the component of electron transport chain or respiratory chain. They are located in the inner membrane. Active transport mechanisms are responsible for the movement of ADP and ATP across it. We shall see this mechanism later in discussion, not in this session but in the next sessions. Negative straining techniques which strain the space around the structures rather than structure themselves, show presence of elementary particles, or sometimes they are called as stocked particles, on the matrix side of the inner membrane. Each particle consists of the headpiece, stalk, and the base. Headpiece, stalk, and the base. So as you can see, here you can see this particle. If we magnify this, it contains of headpiece, stalk, and the base, which is bonded to this membrane. We shall see the magnification of all of these diagrams later. We shall see them as we are continuing with the discussion in our topic of respiration. So the head is associated with ATP synthesis and is a coupling enzyme. The head contains ATPase, which is a coupling enzyme. So here head ya stocked particles inahusika na ATP synthesis. So coupling enzyme ATPase, which link or couple the making of ATP to the respiratory chain. So as we know, ATP is the enzyme which catalyzes the combination of ADP with the phosphate to form ATP. So at the base of the particle, and extending through the inner membrane are components of respiratory chain itself. So at the base of the stocked particles, we have the components of the respiratory chain itself. Now the mitochondrial matrix contains most of the enzymes of the Krebs cycle. Fatty acid oxidation also takes place here. In addition, mitochondrial DNA, RNA, and 7 ribosomes are present. Now, this question we have already discussed about the diameter of the mitochondria. Now, this is the diagram of mitochondria, as how I have already explained to you. Presence of different structures, and we will elaborate them in detail later when we are going to continue our topic of respiration. Now let's move to see the adaptation of mitochondria, because it's one among the questions which is asked in the topic of respiration. Mention the adaptation, or explain how mitochondria is adapted to its function. So when we are studying about the adaptation of mitochondria, I have told you several times that adaptation is how an organelle has different features which enable it to perform a certain function. So you just mention the feature and then you say how it is adapted, or how that structure support a certain function. So for example, presence of appropriate enzymes which catalyze the reaction of Krebs cycle in the matrix, that is adaptation. Then inner membrane, it is folded to finger-like projections called cristae. What are they for? To increase the surface area for electron transport chain. Then the matrix is granular and electron dense. Why is this important? Being granular and electron dense, this will help with the efficient working of the enzymes. Number 4, presence of the permeable membrane. What are they for? Permeable membrane, what are they for? Permeable membranes, they allow the passage of raw materials for respiration. Also, they allow the removal of the end product of respiration, such as carbon dioxide. Also, number 5, mitochondria has stocked particles which contain ATPase. What is that for? ATPase then to catalyze the reaction for formation of ATP. ATPase for synthesis of ATP from oxidation during respiration. Then number 6, they have 7 ribosomes. What are they for? As I told you, the 7 ribosomes, they are protein synthesizing machinery. So they are used in the protein synthesis within mitochondria during its self-replication. Number 7, they have circular DNA. What is it for? Circular DNA is the genetic material, so it carries instruction for different things in mitochondria to occur. So they have circular DNA which enable mitochondria to replicate itself. From there, number 7, mitochondria is covered by 2 membranes so as to separate it from processes taking place in cytoplasm, so as to separate it from processes taking place in the cytoplasm. So this marks the end of our session about the gaseous, about the mitochondria, respiratory substrate, and the mitochondria. And in the next session we'll start our discussion about cell respiration. So I hope you have enjoyed the session. If you need the notes for this session, you can just go to Play Store, go to Google Play Store, download the Kipaji app, and then search for my notes. You will see the notes they have written as Dr. Mlelwa, and then you will see our logo. Which is Dr. Mlelwa Lecturers. So you can search for notes, and they are sold at a very low cost. Ni 2,000 tu ya Tanzania. Kwa utaiona logo yetu hii hapa. Hii logo utaiona pale juu kwenye notes. Na ukishaiona hii logo pale juu, you can know kwamba this is actually Dr. Mlelwa. Kwa lipia hapo, lipia utazipata hizo notes kama zilivyo hapa ninafundishia. So in the next session we start cell respiration. Thank you everybody, and let me wish you nice studies.